Hi there, my name is Arjun Krishnaswamy and I run a research lab at the Life Sciences Complex at McGill. And our interest is to learn more about how the brain becomes wired and how its wiring, which is really the connections among neurons called circuits, give rise to a wide range of our abilities, spanning all the way from reflexes through sensation and motor control, perhaps all the way to more cognitive kinds of things. Now, there's a number of open issues about these structures, particularly how the neurons construct them. And for a lot of our studies, we examine the neural circuits in the earliest step of the visual pathway of laboratory animals, and this is the mouse retina and its immediate targets within the brain. There's a bunch of recent work that suggests that the eye isn't a camera. It doesn't just take a picture of the world and send it to the brain, but rather the eye contains a number of neural circuits, and each of these circuits appears specialized to detect a particular aspect of the visual scene. And so maybe the retina would look at a painting like this and then decompose it into a bunch of different versions of this image, each of these containing emphasis on a certain aspect of a painting, which reflects the feature detecting abilities of certain circuits in the eye. Understanding how these circuits work is therefore critical to figuring out how those functions relate to wiring patterns between the component neurons, and then also understanding where the blueprints are. So we also look at the genes within these cells and try and understand what addresses them to one another. Now, for a lot of these studies, we build custom instruments in my laboratory. So we build special microscopes called 2-photon microscopes, which employ an infrared laser, which is projected onto the surface of the retina and then used to evoke fluorescent signals from individual neurons so that we can go in and record electrical signals from them or deliver special dyes to these cells that allow us to image their neural activity. So here is the kind of data that we collect using these kinds of microscopes. And so briefly what we've done is to take a retina, put it inside a dish, put it under this microscope, and then we're showing patterns to this using a small projector that can show movies to the retina. And then we're going to measure neural responses from these cells. The cells are shown here, and each of them is one of these white dots. And truth is, they're actually green. They're labeled with a special dye that glows brighter when the neuron becomes active. And over here, when I play the movie, you're going to see the responses of the same cells to a stimulus where we flashed a big square all around this region of neurons. So I should just say that each of these guys views the world through the photoreceptors that live above it. Through an intervening layer of interneurons. So we're going to show a big square to this group of cells, and then we're going to figure out which ones of them, when the square flashes bright or turns dark. And as you can see, the cells go on and off and on and off and on and off and so on and so forth. Gathering this kind of data is essential to understand how the retina and its circuits work and how they analyze the visual scene. And it's made possible by the presence of a powerful, cost-effective 920 nanometer fiber laser from MPB Communications. Their staff is friendly, knowledgeable, and attentive to any need that arises. The installation was straightforward, and the systems are really plug and play. I've been very happy and fortunate to work with them and collect this kind of information. And most of all, the quality of these femtosecond lasers are really fantastic, and I would recommend them to colleagues without hesitation.